Okay, I guess we're ready here. So, I've been looking at the discussion board and searching my own soul, of course. And I see there's great confusion about the genome engineering project and what's extra credit and how to get it done and all that. And I agree I'd probably be confused too. So I just took a hard look at it this morning, and I've looked at the graduate section of the course, and I have put up a revised syllabus which provides more opportunities for extra credit. And I've made it easier to understand how the genome engineering project will influence your grade. For graduate students, they are required to do the genome engineering project. For the undergraduates, the PCB 4522 people, it's an extra credit option. So there are two levels of participating in that genome engineering project. The first level will give you 3 points or 3% extra credit as an undergraduate. And that is simply go to the videos and submit exactly what I submit. Follow along with me. A lot of people have trouble doing that. If you can do that, 3 points extra. Like if you have a 87, that'll give you a 90 for the final grade. It's very powerful. Just submit what I'm submitting. So I don't want you to give me a FASTA file or a nucleotide file, because I can't remember what's the sequence of these genes. The most important thing is you have it annotated, that you explain what you have done. So that's what I'm looking for. Do you understand it? Could you go to the sites and reconstruct what I did using the same genes? Now, if you want to get 5%, that's half a letter grade, then you're going to have to do at least one original construct of the 3 that are required. So look at the scheme. It'll take a little thinking about what you'd like to substitute. There's some easy substitutions, but you'll have to get your own promoter or make your own hairpin construct, or there are 3 different constructs there. If you make your own, then you have potential to get up to 5% extra credit on the genome project. So you can get 3% or 5%, up to 5%. That's the way I'm thinking about it now. I'll update the syllabus probably tonight and let you know, and I'll give you an upgraded grade calculator so that you can consider what your options are in terms of taking the optional final. Question?
And how is this different from the part 1, part 2, part 3 assignments?
They are mandatory. They are built into the 100%. And so each one of those gives you 3 points that are part of the course, which will add up to 100% without any extra credit. Okay? The graduate students have some options about extra credit. I may expand some of those for you also. For example, for the graduate students, I've given them the option to prepare a short PowerPoint presentation on a molecular biology topic of their choice, and I give them some choices that have been popular in the past, but it could be anything that's molecular biology, not clinical, not enzymatic, not biochemistry, but molecular biology, gene expression, or DNA replication or repair, the topics we've covered. And the third option I'm just kind of throwing out there, I'm calling it molecular art. You saw a little crazy thing I did. If you look on discussions, I tried to make myself some Rec ABC. It turned out to look too weird for me to present publicly. So I stood back and said, what is that? I'm not sure I want anybody to see that. But there it is. I made a little clay model, and then I took pictures of it, and then I sketched it and colored it and had lots of fun with it. And you can take it from various shots, and I can have it emerging in a jungle clearing and being with people around worshiping it. All sorts of possibilities. Rec BCD. Whoa. Yeah, it's pretty heavy. Make your own, the pre-initiation complex. Make a model. You can get up to 5% extra credit for that too. You can only get 5% extra credit no matter what path you take. So it's not going to be like the whole course is extra credit, because that'd be way too easy and way too much fun. You wouldn't value your degree. So 5% is possible. I'll think about this and give you those opportunities so you can maybe, we can sort of lighten it up and have some fun here at the end, and so you won't end up watching all your friends go to medical school. You can have your own GoPro and take it to medical school with you. You can be the guy, the person. Okay, so any questions on this nebulous thing called syllabus? This nebulous thing called instructor expectations for the students. This whole thing that should be in concrete by now, but is still a little mushy. Okay, I apologize for the mushiness. The course has always been a work in progress. Each year, it's a journey, this course. Okay, so let's do a little backtracking here. One of the things that I told you about was that, whoa, look at, for all these, 138 years, I'd been teaching, and we thought that the mitotic chromosome was really a higher order folded structure. I remember when I took geology as a biology major. I was interested in all the ologies. It was a free elective. I was not going to take, medieval armor. I decided to take another science. I'm interested in rocks. Everybody has a little rock collection. Where's the water table? How are mountains built? And the new idea that this old mustached professor told us about was that what was being debated in the literature of the time, and we were lucky to be alive when this was presented, was the idea of plate tectonics. Yeah, pretty heavy. And I'm going, whoa, I just love college, sitting in class and getting all that information just blasting by me. And so this is not quite that kind of moment, but chromosome structure. We're finally getting it figured out. Maybe, but this is the current view of how it works. What's it all about? All these 30 nanometer, 50 nanometer, 200 nanometer, and so on, until finally you got a beautiful chromosome. Seems to be totally made up. Artifact of the purification procedures which strip away a lot of proteins, and you see arrangements of chromatin that are probably not in vivo. So we talked about the way that heterochromatin and euchromatin seem to have the basic underlying structure. You have beads on a string, and then these little beads can clump together depending on the proteins that the nucleosomes are decorated with. They can form little globules, if you will, of like proteins where they dissolve in each other. So no difference between euchromatin and heterochromatin except the density of this sort of tangled mass. But I had trouble going up to the next step. From heterochromatin to the mitotic fully condensed chromosome. Is there a 30 nanometer fiber? Are there higher orders of folding? It just seems like there's something else going on as you go into that fully condensed state of the mitotic chromosome. And so I hedged my bets here in class. I said, I'm going to have to do a little more reading. Well, I've done the little more reading, and the answer is it's the same. It's the same organization as you go further up. And the clue, in hindsight, was what we were hoping was a clue, is the lamp brush chromosome. The loops. It turns out it's loops upon nested loops upon loops. And this is an article, actually it's a little movie that I discovered this morning preparing the lecture. And so it won't take very long. So hopefully it'll play. Hope. Never hope. If I can copy this into memory. So made of these things. So here goes. So this forms the axis, and this nucleates around the proteinaceous scaffold of the chromosome. It's a docking post for the base of the loops, a nested loop. There, and this is where the kettle drums would break in. Okay, so I have inserted that slide and link in the chromosome part 1, and I'll probably put one more slide. I read one more paper. It would just be a separate study that this model was actually based on. But there are a series of papers that are coming out that seem to confirm this view. So it's pretty exciting for me. Okay, let's go to, where were we? We were going through the parts here. We'd done eukaryotic transcription. We did part TF2D almost, and then we backtracked and then did 1 and 2 last time. But we didn't quite finish part 3, so now we're going to go to the one that I jumped on too early. And this one, you remember TF2D? So we talked about in parts 1 and 2 the overall strategy for building a promoter in a eukaryote for Paul 2. We talked about Pauls 1, 2, and 3, and the common components, the fact that TBP was a part of the commitment factors for each of those. And we talked about enhancers and their location, the fact that they are different than the core promoter, and we began to talk about elements within the core promoter for Paul 2. What we're going to do now is continue our discussion of the basal transcription factors that nucleate at the promoter, at the core promoter. Those that bind at the core promoter, which is within roughly 100 base pairs of the start of transcription, need to bind there for every gene, every promoter that's regulated by Paul 2 will have these basal transcription factors recruited and binding there as part of the building of the pre-initiation complex. And the first to arrive, and the first we're going to discuss, is transcription factor 2D. And we've talked about how it's TBP, which is Tata binding protein, and its friends, the TBP associated factors. And we talked about the roles that these that TF2D does. It recognizes the core promoter. It's a target for recruitment by transactivators that are bound at the enhancer or upstream elements. And then it in turn recruits other basal factors, and one of the TAFs has enzymatic activity. That's the largest, which is TAF1. We've talked about the idea that most promoters have no Tata sequence. That doesn't seem to be anything for Tata binding protein to do. There's no Tata, and there's not one for Paul 1 or for Paul 3, except for that third group of Paul 3. And so, who needs the Tata sequence? Where did that come from? It comes all the way from Archaea, and you can immediately recognize the similarity. Where did Archaea get it? Well, Archaea seems to have gotten it from the eubacteria, because that looks like the -10. How does it differ from the -10? Location, 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 right? It's about a little over 20 to -30, -20 to -30 upstream from the start of transcription. So the promoter doesn't actually melt in the Tata in a eukaryote. The melting overlaps the Tata in bacteria, but not in eukaryotes. It's used simply to recognize that this is a promoter in eukaryotes and in archaea. So it looks like eukaryotes have gotten away from the original consensus binding of the TATA as a function for the TATA binding protein. 90% have no TATA, but those that still have a TATA are a special class. They're genes that can be induced, not constitutive housekeeping, chugging along, but ones that are induced by environmental stress, abiotic or biotic, which means pathogen attack, a pathogen response, a heat shock response, for example, or some developmental signal, developing a particular tissue in maybe development of a mammal or of a plant, developmentally cued regulatory events. So let's look at some of the detail here of these TAFs. Some of you in the gene structure project were assigned a TAF, and it'll have a crazy name like RNA Pol2 promoter factor or something. You'll never guess it's a TAF unless you look down the list of things in the BLAST sequence and you'll start seeing TAF9, TAF9, TAF9. That's what it is, it's TAF9. When they first annotated it, they didn't fully realize what it was. So the first function, the TF2D recognizes components of the core promoter. And here you see, we know that TBP, if there is a TATA, it will be recognizing it. But let's assume there's no TATA, because that's how most of them are. How does it know it's at a promoter? Well, these other elements, the downstream promoter element and the initiator region, are recognized by specific groups of TAFs. TAF 1 and 2, they are the two largest. They together recognize the initiator region. TAFs 6 and 9 together are able to recognize a downstream promoter element. So you see these TAFs are really plug-in information modules that give it the brains to recognize these promoters that have no TATA. How about 6 and 9? 6 and 9 also are targets of transactivators. These transactivator proteins, these are bound to cis elements outside of the core promoter. And they have a DNA binding domain, these transactivator proteins, and they have regions that are called activation domains, and they are really regions of affinity for targets. Some of the targets would be the TAFs, as they try to recruit TF2D to the core promoter. So TF2D is not scanning necessarily, it's being grabbed out of the sky and slammed down on the promoter. The handles, the point of contact, are TAF 6 and 9. That's the second thing TAF 6 and 9 is doing, right? The first was to recognize the downstream promoter element. Here, the same TAFs are involved in as targets for recruitment. Oh man, tell me this can't really be on the exam. I can't definitely say that. Can't make that statement, but probably. What you'll need to know, probably there's a question pool or group that will ask you sort of to match up the function of these TAFs. It's horrible. This is like, there are lots of horrible things in this course, and this is one of them. Hopefully there won't be too many more. But this is a thing you're just going to have to commit to memory. If you looked at the How to Study guide, this would be a slide you'd put a little check beside. This is a slide I hate. I'm going to have to go back and study that. This perhaps is a big gaping gap in your knowledge. Fill it. And nobody was born with this information. Okay, TAF 6. It contacts other basal factors. You know, looking down the list of what TF2D does, it's a target for recruitment, it recognizes a promoter, and then it recruits other basal factors. It looks like TAFs 6 and 9 have involvement in all of those roles. The last one having enzymatic activity in it does not have that. So what does it reach out and touch? TF2E, that's the other basal factor, and E has 2 subunits, and it recognizes the alpha subunit, which is the largest. This alpha subunit, TF2E, is ancient. It goes all the way back to Archaea. Archaea has TF2E, believe it or not. TAF6 grabs it. TF2F, the small subunit of that, is also a target. RAP30 is another name for that. TF2F, that's RNA polymerase associated protein 30. That was its old name. So TAF6 is recruiting 2 of the basal transcription factors, that many more. E, H, F is the red, A and B. We've talked about B, we talked about A, we don't know much about A. D is this TBP with all the TAFs, and you see TAF6 is making contact with the small subunit of F right there. It's also making contact with the large subunit of E. TAF9, 6 and 9, they've usually been a pair, and now you see they have separate affinities also. Here you see TAF9 is making contact with TF2B. 9 making contact with B. So 6 is making contact with E, and 9 is making contact with B. TAF4. TAF4 is recruiting TF2A. So you can see how it makes sense for TF2D to arrive first, because it makes possible the recruitment of all the other basal transcription factors, either directly or indirectly. TAF1. When will this list stop? Pretty soon. But not soon enough, right? TAF1, this is the largest one. TAF1 is like 250 kilodaltons. It makes contact with the large subunit of TF2F. That's called RAP74. So already we know a lot about TF2F. It has 2 subunits, an alpha or a large subunit, which is RAP74, and a smaller one, which is called RAP30. Okay, surely this is oh, there's another one. I think this is the last. Okay, TAF5 also makes contact with that large subunit of TF2F. What? I don't know what to say. I don't know what to say. I'm going to have to go back and look at exam 4 because I don't know. This is horrible that I wouldn't put this on the exam. I don't know whether it's on there or not. I'm going to have to make sure that, I don't know. I'm so surprised. I don't know who does these slides. Okay. Let's dig down into TAF1 a little bit more, because I know you're all very curious about TAF1. That's the one with the enzymatic activity. What kind of enzymatic thing would you want to do with a TAF? Well, here you see its structure, and you go, really? And you're going to tell us this is not going to be an exam either after you've gone through it in detail and we wasted all the brain gigabytes for nothing? Okay, surely this will be on there. These are the 3 activities. It has a kinase activity, and it's split into 2 parts. A kinase adds a phosphorus group. It has a histone acetyltransferase activity. That's interesting, isn't it? That's chromatin modification. That modifies the tails of nucleosomes and it acetylates them. Acetylation of a histone tail is generally associated with activation. So you've got something that's recruited to a promoter, and it seems to be promoting, keeping the chromatin in an open configuration with hat activity. UBACA? What is this? Is like a Star Wars thing, right? UBAC. That's ubiquitin acetyl conjugating activity. Let's see how this goes here. There it is. Ubiquitin activating / conjugation activity. This is pretty interesting. Some of you are aware that the proteasome in eukaryotes is a big protein degradation machine. You know those things that go down the highway and rip up the old highway so the new highway can be there and spit out the gravel? The proteasome is like that. It takes proteins and is constantly funneling them in and grinding them out and spitting them out as amino acids. And how does it know which protein to degrade and which ones to let alone? It's a molecular tag that's put on the lysines. Ubiquitin. It's a ubiquitin tag. It's a little peptide that is in a repeat. If it's polyubiquitin, same repeat over and over again. And it's covalently linked to lysines, kind of hanging off like strings. That targets it for degradation on the proteasome. But this doesn't give that kind of signal. This puts on one ubiquitin tag. It monoubiquitinates. It's a fun word to say. Monoubiquitination. It's very staccato. You feel like you've said something. You can make your cat's eyes roll back. Monoubiquitination. When you add a single tag, it doesn't target it for degradation on the proteasome. It simply inactivates the function. It just gets in the way. And what is the target for this? Histone 1. Remember, histone 1 is the one in the old days was required to condense chromatin to the higher levels to make the 30 nanometer fiber. It's probably involved in chromatin condensation somehow, but not to make a 30 nanometer fiber. Maybe that's one of the things that help make these clumps of nucleosomes. But you see, by polyubiquitinating histone 1, you've prevented higher orders of folding from happening. Again, promoting an open, non-condensed configuration of the chromatin. So you have two agents here, enzymatic activities that are promoting maintenance of an open chromatin configuration. We're not quite sure the significance of the phosphorylation of TF2F. That is still being worked out. So, three activities: kinase, HAT, ubiquitination, activation, conjugation. There are other things in here. There's a zinc domain for binding, some bromo domains, but look at the N-terminus. It's something called TAN 1 and 2. You know these anglerfish? They look like a pile of garbage, and the tongue is like a worm. It's on a pole just flirting around. To me, that's what TAN 1 and 2 are. And who is it trying to attract? Who's going to snap it? Our friend TATA binding protein. Because on the underbelly of TBP, that's where it's looking for that minor groove for the TATA so it can bind it. This little domain there, TAN1, is a protein mimic of TATA, which is DNA. The charge distribution on TAN1 looks like a promoter element, and it's just darting around up there, and TBP snaps it and thinks that it's just stepped into paradise. It stops looking for a promoter element and now is perfectly content to bind on a TATA-less promoter because it's all happy. That is on the end of the largest TAF, it just bends around and fits right underneath the DNA binding domain of TATA binding protein. It masks TBP, so TBP is no longer searching for the TATA sequence of the promoter. TAF1 takes care of it. Puts on those funny glasses where it looks like it's in an ice cream parlor. It's not looking for ice cream anymore, it's found it, it's arrived. Oh, TAF1. Oh me. The TAF1 is conserved. The domains are present in whether it's a plant or whether it's yeast, but sometimes you can split the protein up into a couple of proteins as you see in yeast, but all the domains end up still being there. Except the plant has an undeveloped TAN1 region, but you don't need to know this for the exam. Okay, we'll get back to that TAN1 domain. There's a slide that shows how it works. But let's take a little detour now and look at how TF2D is involved in regulating the building of the pre-initiation point. It serves as a checkpoint. So here you see one of the TAFs, TAF7. TAF7 has two configurations. It's either bound to TAF1 and 7, or TAF7 is bound directly to TBP. If this region here is accessible, then it will prefer to bind to TAF1. So let's see what happens there. When RNA polymerase actually arrives at the pre-initiation complex, that acts as a signal for phosphorylation. TAF1 phosphorylates itself. When it does that, it causes TAF7 to lose affinity. TAF7 now transfers over to TBP. When that happens, notice that you have exposed the ubiquitination, the monoubiquitination activity. That's so you don't do that too early, apparently, who knows. And you can now have HAT activity. HAT activity also is masked by TAF7. Both of those things are prevented before the arrival of RNA polymerase itself. Arrival of polymerase triggers autophosphorylation, which gets rid of this masking agent, TAF7. So now these other two enzymatic activities can be expressed. So this is some of the details of the TAN1. I've already given you the story. What you see here is this is that domain at the end, the TAN1, that mimics the structure of DNA. It mimics the TATA in the promoter, and here you see it interacting with the underbelly. See the two stirrups? This would be the C-terminal stirrup because you see the two ends over here. It interacts with the DNA binding domain of TBP. And here you see a space-filling model showing that. This is the DNA, and this is the protein mimic. Not bad for a protein mimicking the topology of DNA. Of course, TAF1 prevents TBP from binding to TATA when it's there. Okay, that's the end of the TAF1 story, I think. Pretty sure. Now we're going to look at something that we thought was going to tell us a lot, but we don't know where to take the story to the next step. It turns out that TAFs 6 and 9, remember 6 and 9 recognize the DPE, 6 and 9 are targets for acidic activators to recruit TF2D, 6 recruits TF2E, 9 recruits, what's that, B? No, 9 recruits. What does 9 recruit? B? B. But there's something else about them. They have within them the histone fold. For 3 and for 4. One looks like 3 and one looks like 4. Buried within them is a perfect mimic of these two ancient histones that go all the way back to Archaea. Does this mean a nucleosome got smart? Did TF2D evolve from an ancient nucleosome that instead of just binding DNA nonspecifically, began to pick up subunits that allowed it to recognize promoters? I don't know. As the guy in the Geico commercial looking for a lug wrench says, maybe. But what? There's more. TAF 12 and 4B, they mimic H2A and H2B. It looks like we have a complete nucleosome there reconstructed within this basal transcription factor. What does it mean? Does DNA wrap around this thing? There are models that suggest that. We don't seem to know fully yet. These 4 histones are what is called the core. They look like an ancient nucleosome. And these are shared by other complexes in addition to TF2D. There are alternate forms of TF2D, all of which deliver or escort Tata binding protein to the promoter. But some escort TBP to the promoter with the mask off, where TBP now is looking for the Tata. So if you want activated transcription, you're going to not use TF2D because TF2D is not going to look at the right kinds of promoters. The kind of promoter you want to look at are those that have Tata. You're going to need a fully unmasked, fully functioning TBP, and the way to do that is to have TBP arrive at the promoter without TAF1. TAF1 has that TAN1 domain that masks. And so these alternate complexes you see here at the bottom of the slide, SAGA and PCAF. SAGA is in yeast, PCAF is in mammals. And these are alternate forms of TF2D, essentially, except they're not TF2D in that they don't have the full complement of TAFs. They don't have TAF1, but they do have these 4 that seem to mimic an ancient nucleosome. Something is conserved about that. Something provides evolutionary advantage. This shows the molecular structure of TAF 6 and 9. If you can remember, see these 4 helices, helix, a long helix, and then another one? That's exactly what TAFs 3 and 4 look like. There are models which suggest that perhaps the DNA wraps around. The models haven't really gone anywhere. They haven't gotten a lot of traction, so all that's still kind of unresolved. So what does it really look like? We have some topological or surface information derived from a Fortran transformation of 2-dimensional electron microscope pictures of the complexes, and it looks like this sort of structure. And it's thought that maybe DNA is wrapped around the center here, like this, because TBP is in the center, and TF2B and A on either side. And again, when I see this, and perhaps all of you now, you're seeing creatures from the Wizard of Oz, those that ride brooms and wear pointed hats. I mean, those guys are talking to me. I'm trying to get that fixed, but every year I see these guys in very animated conversation among themselves at the bottom of the slide there. There are multiple forms of TF2D. There are multiple configurations or conformations of it. We don't fully understand them at the moment. This is some mapping information where antibodies to TF2A and B were used to determine the spatial arrangement of the basal transcription factors within this TF2D complex. You see TBPs in the middle, B on one side, A on the other. Fun facts. They're not really fun, are they? TF2D is often used at promoters that lack a tata motif, constitutive genes. That's 90% of them. Note TBP can function with complexes other than TF2D, and that's SAGA and PCAF, which you've talked about. Many of the TAFs are involved in developmental processes. You can delete some TAFs and you won't kill the organism. And then many of the TAFs are in these alternate complexes, these SAGA and PCAFs, but the ones that are especially important are the histone fold TAFs. And you remember 6 and 9, 4 and 12B. In yeast, 90% of the genes have promoters that are primarily depend on TF2D. It's the main workhorse, and that's because most promoters have no tata sequence in their promoter. That's why you need a smart complex that has all those other subunits that know how to recognize a tata-less promoter. Limited capacity for induction. Remaining 10% rely on SAGA. In mammals, that would be PCAF. Stress inducible, wound inducible, developmental inducible. Okay. Alternate complexes. How much? We've got 4 minutes. The alternate complexes, you're going to need to be able to distinguish between the SAGA, the PCAF, and the TF2D. What do they have in common, and how are they different? So let's look. All of them, all of them share a subunit that contains HAT activity. In TF2D, it's TAF1, right? That contains all the enzymatic HAT, TAF1. There are things, subunits that contain HAT in the other complexes too. GCN5 is the name of the HAT-containing complex in SAGA. GCN4, just to complicate life, similar name, but this is a transactivator that binds at the promoter and recruits the TBP associated factor, whether it's D or SAGA, to the promoter. It turns out it's specific for SAGA. This is an example of a transactivator that cannot recruit TF2D. It specifically pulls down and recruits SAGA. So you'd find this at inducible genes, wouldn't you, as a transactivator? This is a Bronze Age picture, very ancient, but it still has validity on exam 4. I hope one of those twirly signs doesn't come up. TF2D, TAFs, and TBP, SAGA, TBP is associated with it. GCN5 is the subunit that contains the hat activity. In humans or mammals, PCAF it takes the place of SAGA. It's essentially the same complex. The subunit that contains hat, I'm preparing you for a specific exam question, multiple choice. Which subunit contains hat? In PCAF, it's a subunit for which the entire complex is named. It's the PCAF subunit of the PCAF complex. And then there are others. And these alternate complexes have varying affinity for TBP. But somewhere along the way, their role is to escort TBP to the promoter. And when you biochemically purify them, they have varying affinities for direct contact. But their role, you have to have one of these to get TBP at your promoter. Why do you need TBP? It doesn't recognize the TATA. Why? What's the insistence that we bring TBP there every time? Because TF2B needs to bind it. And TF2B has to insert the N-terminus into the active site of the polymerase itself to assist in the melting and to have this interplay with the nascent transcript. P300 CBP, these are very similar, but they are distinct, so they have different names, and we'll talk about P300 and CBP in terms of steroid receptors. So from this, you should know that TBP is either tightly bound or recruited by these alternate forms. You know, these are sometimes called co-activators. Although I don't like that name because these are basal transcription factors. These are directly involved at binding and recruitment events at the core promoter. But because they have hat activity, some people call these co-activators. Know which subunits are in common, and that would be the 4 core TAFs, the ones that look like a mimic nucleosome, 6, 9, 4, 12B, and which one contains the hat activity. PCAF, GCN5, TAF1. Okay, into part 3. This is the end of the lecture. Worked out just right. So see you on Thursday. And be looking for the update in the syllabus so you get all the points and the grade, extra credit sort of cleared up.